This is critical thinking in urinary anatomy lab, and here are the questions. So here's the first one on certain important cells. A second one which shows you an X-ray, but you have to identify the structure first and then answer the question. And then here is an image. And let's go to the first question, which where the question asks you, what cells does the arrow point to and what is the function of these cells? So let's look at what the arrow is pointing to, and you can see that this is a blood vessel. This is, as you can see from this, here the diameter is narrower, here the diameter is wider. So this is the afferent arteriole, and here this is the efferent arteriole. So these are cells which are present in the afferent arteriole, and these cells are called juxtaglomerular cells, or also called JG cells. And the way you'll remember this is this area, this capillary network here, is called the glomerulus, and juxtaglomerular means close to the glomerulus, and you can see that these cells are close to the glomerulus. So these are known as JG cells. So that's the first thing you will identify, and then you will talk about the function of these cells. So let's look at the choices we have here. So the first choice says macular densa cells. As I said here earlier, these are called JG cells because they are present close to the glomerulus, which is made up of capillaries. Macular densa cells are these ones, and they are seen in the distal convoluted tubule. So hence it cannot be this answer. In a question such as this, you will notice that the first part of the question before the colon is answers this part of the question, and then the second part of the question would answer this part of the question. Now macular densa cells, even if you did think of this as a choice, if you look at the second part of the statement which says sense blood pressure in afferent arteriole, as I said, macula densa belongs to the distal convoluted tubule. So it's not going to be able to sense blood pressure. Blood pressure can only be sensed in an arteriole. These cells are not part of an arteriole, so in any case this statement is wrong, or this answer is wrong. Let's look at the next one, which says JG cells. So that part is correct. JG cells is correct. But then the next choice is sense solute content. Now if you look at it, solute content is the content which is present in the filtrate. So this has to lie in the nephron, in one of the parts of the nephron. And it cannot lie, the filtrate does not lie in the arteriole. Blood lies in the arteriole. The filtrate lies in the distal convoluted tubule. So hence JG cells cannot sense solute content. So this part is wrong, hence this choice would also be wrong. Let's look at the third choice, which says mesangial cells. These mesangial cells, these are cells which are present here. So in between the arterioles and this distal convoluted tubules. So these are the cells. So hence, as we can see here, these are not the cells that the arrow is pointing to, and the function of these mesangial cells is indeed to pass signals between macula densa and JG cells. Let's look at the fourth choice, which says macula densa cells and to sense solute content. The function of macula densa cells is to sense solute content, because solute is what flows through the distal, or filtrate is what flows through the distal convoluted tubule, and that will have solutes in it. But the arrow is not pointing to these macula densa cells, hence this is not the correct answer. The correct answer is this one, which says JG cells. So these are the juxtaglomerular cells, you can see close to the glomerulus, and they do sense blood pressure because blood is flowing through the arteriole. Therefore, cells present in the arteriole will be able to sense blood pressure. Cells which line the distal convoluted tubule will not be able to sense blood pressure because no blood flows through the distal convoluted tubule. Filtrate is flowing through that, so hence these cells which are macula densa, they will be able to sense solute content. So it's very important for you to think critically when you're answering questions on what the cells will do, and based on where they are present you should come to the right choice. Now let's look at the next question. which asks, why is it important to know the location of normal constrictions in the structure the arrow points to? So this is a special X-ray which is known as an intravenous pyelogram, or also called often called intravenous urogram, because it outlines the urinary tract, and the arrow is pointing to the ureter. And if you look at this X-ray, these are actually, this part here is showing you the calyces of the kidney, this part here is the pelvis, this is the ureter, and then this is the urinary bladder. So these are the parts up here, so the arrow is pointing to the ureter. So it asks you, why is it important to know the location of the normal constrictions in the structure? So the normal constrictions in the ureter, and of course you must know the normal constrictions first, which is where the first one is here at the pelvi-ureteric junction, where the pelvis becomes the ureter. The second is where the ureter crosses the pelvic brim around this point, and the third one is where the ureter enters into the bladder. So these are the three constrictions, and the reason why you want to know that is, let's look at the choices. To determine where a calculus is likely to get lodged. This is the correct answer, because if somebody had a kidney stone, which is what a calculus is called, since the ureter is normally constricted at these points, if you look like this, so there's a constriction, and then again there's a constriction like this. So if there's a stone, as the stone gets passed down, it's likely to get caught at points where the ureter is narrow. So this is the correct answer to this question. Let's look at the other choices to make sure they are not better than this one. So the second is, these are areas which lack a blood supply. This is not true. It's just that there is a normal constriction over there. It's not that the ureter lacks a blood supply. So this is not true. And the third, which says to be careful passing catheter while performing an intravenous pyelogram. As the name suggests, intravenous, that means actually you don't pass a catheter into the ureter. So you don't pass something through here into the bladder and then into the ureter going up. In order to take this kind of an X-ray, you inject a dye into a vein. That dye goes into the bloodstream and then is filtered through the kidney, and then you can see as it's filtered through the calyces, passes down the ureter and then into the bladder, you can see the outline. So you don't need to, so a catheter is not passed, so hence this is not the correct answer either, and this is the correct answer to this question. In fact, it's a good idea to also know this, because you'll be able to distinguish a normal from an abnormal intravenous pyelogram, because if you were to see a constriction in this area, let's say there was dye missing in this area, you would know that it's because of the constriction, and maybe there's very little dye and that's why you can't see it. Now let's look at the third question, which asks, which of these structures leads to a peritubular capillary plexus? That's the first part of the question. And which of these structures opens into a minor calyx? So let's first identify all these structures. So A, as you can see here, is the efferent arteriole. B is the afferent arteriole, and the way you will identify it, if you remember, the afferent arteriole comes off from the interlobular artery, or also known as the cortical radiate artery. Now this is the arcuate artery, and this artery here is the interlobular or the cortical radiate artery, and arising from this you can see this B coming out. Then it goes in here, forms the glomerulus, and the efferent arteriole exits from the glomerulus. It has no connection to this cortical radiate artery. So you can see it has no connection. So therefore this is the efferent and this one is the afferent. Where you see the afferent connected to this interlobular or cortical radiate artery, that would be the afferent arteriole. Now all of this is the peritubular capillary plexus. C is the loop of Henle, so this is the loop of Henle, and D is the collecting duct. So now it says, which of these structures leads to a peritubular capillary plexus? As you can see from this diagram itself, you can see that this A is the one which leads to a peritubular capillary plexus, which is the efferent arteriole. And then it asks, which of these structures opens into a minor calyx? Obviously, none of the arterioles will open into a minor calyx. So that means the second part of the question, anywhere where you have an A and a B, you can exclude that choice. So this one can be excluded. So it has to be one of the tubes. So it either is the loop of Henle or the collecting duct. Now the loop of Henle does not open anywhere. It goes back up and becomes the distal convoluted tubule. It is the collecting duct which opens into a minor calyx. So it is D which opens into a minor calyx, and it's very important for you to know the order. So the collecting duct will open into a minor calyx, and the point actually where it opens is this area is the apex of the pyramid, which is known as the papilla. So the collecting duct opens into a minor calyx. The minor calyx, you have a couple of minor calyces, they open into a major calyx, and then the major calyx opens into the pelvis, and then the pelvis becomes the ureter. So you have collecting duct going into minor calyx, which opens into a major calyx. The major calyx goes into the pelvis, and the pelvis then leads into the ureter. So we've determined that A is the one which gives rise to the peritubular capillary plexus, and D is the one which opens into a minor calyx. So it would be A and D. So this would be the choice, the correct choice. And just to show you, the same picture is what is seen here, and I'm showing you this. So if you look at this diagram here, you can see again it is very similar to what you saw earlier. So these are the arcuate vessels. This is the interlobular artery, and here coming from the artery, this branch here is the afferent arteriole, leads into a glomerulus, and then you can see this one which has no connection with the interlobular artery. This is the efferent arteriole. This efferent arteriole gives rise to this whole bunch of capillary plexus, which is called the peritubular capillary plexus. That capillary plexus finally leads into a vein which exits and gets into the interlobular vein. Then you can see here in yellow or light yellow, here seen better here. So here again you can see this is the afferent arteriole, this is the efferent arteriole. Notice how the afferent comes off from this interlobular artery. Here's the efferent arteriole. This is the Bowman's capsule, glomerulus. So this is the proximal convoluted tubule. This is the distal, this is the loop of Henle. This here is the distal convoluted tubule. So this is the loop of Henle. This part here is the loop of Henle. So let me just do that again. So here is efferent arteriole. This is afferent, the glomerulus and Bowman's capsule, proximal convoluted tubule, loop of Henle. Here is the distal convoluted tubule, and many of these open into the collecting duct, and this collecting duct is the one which will open into the minor calyx. Very often students, when asked what opens into the minor calyx, they'll say the major calyx. The major calyx doesn't open into minor. The minor is the one which opens into the major calyx. It's the other way around. So this should now explain how you identify the arteries and how you identify the various other parts of the nephron.